Dear students, today I am going to talk about bio-based plastics and fibers. The increase in population, industrial advancement and environmental pollution has evolved into a full-fledged global hazard. Today, packaging material account for more than 60% of municipal garbage, the majority of which are non-biodegradable synthetic polymers. It is indeed a clear indication that biodegradable packaging alternatives for ecological sustainability are urgently needed. Because of benefits like biodegradability, cheap cost and accessibility, biodegradable polymers are viable solution for production of eco-friendly products. In recent years, there has been a surge in curiosity of film manufactured using green and biological materials. Polysaccharide, which includes starch, pectin and cellulose, can be used to create recyclable and consumable films. Starch is the most abundant and an important class of biopolymer. Starch is a carbohydrate polymer that may be produced at a minimal price from a wide range of crops. Naturally, starch is made up of combination of amylose and amylopectin. Amylose is a straight molecule comprising alpha-1,4 linkages. Amylopectin is a branch molecule with alpha-1,4 and alpha-1,6 linkages. Amylose is vital in film making since it is responsible for starch's ability to create films. However, biopolymer derived from starch-based film is brittle and has low mechanical qualities compared to non-biodegradable plastic films, which are used in food packaging industry. However, biopolymer derived from starch-based film is brittle and has low mechanical qualities compared to non-biodegradable plastic films used in the food packaging industry. As a result, starches are changed to improve their effectiveness in various applications. According to research on starch-based films without additives, these films are brittle and sensitive to moisture. As a result, the use of plasticizers overcome the brittleness of starch films and enhance their flexibility and processing. Water, oligosaccharide and lipids are some of the plasticizers found in films. Dear students, let us discuss about what is bio-based plastic. Bio-based plastics are fully or partially made from biological resources rather than fossil raw materials. Bioplastic manufactured from bio-based polymers stand to contribute to more sustainable commercial plastic life cycles as a part of circular economy, in which virgin polymers are made from renewable or recycled raw materials. Thus, bioplastics are plastic materials produced from renewable biomass such as vegetable fats and oils, cornstarch, straw, wood chips, sawdust and recycled food waste. Some bioplastics are obtained by processing directly from natural biopolymers, including polysaccharides, for example, starch, cellulose, and proteins, for example, soya protein, gluten, and gelatin, while others are chemically synthesized from sugar derivatives, for example, lactic acid, and lipids, oils, and fats, from either plants or animals, or biologically generated by fermentation of sugars or lipids. In contrast, common plastics are derived from petroleum or natural gas. Bioplastics have advantage like dependence from fossil fuels as a raw material. Life cycle analysis show that some bioplastics can be made with a lower carbon footprint than their fossil counterparts. For example, when biomass is used as a raw material and also for energy production. Dear students, let us discuss plants as a source of starch. Plants and starch. Starch is classified into three types based on its breakdown by amylase. The first one is fast digestible starch, the second is slow digestible starch, and the next is resistant starch. There are several health benefits of resistant starch. While delivering a sense of fullness, it escapes digestion and enters the colon, where it ferments and functions as a prebiotic for the bacteria that aid digestion. Plants also have a good concentration of resistant starch. Extraction of starch for bioplastic. The method of extracting starch depends on which source the starch comes from. Wet milling is used to extract millet starch via one of the three water steeping procedures like acid steeping, alkaline steeping, or a combination of these two methods. In these techniques, the entire kernel is steeped in aqueous solution for a prolonged period of time. Choosing a process that offers a better yield of starch with a little protein contamination is crucial. The alkali steeping method is regarded the optimum approach for millet starch extraction. The procedure comprises the use of sodium hydroxide solution and neutralization by dilute acids such as hydrochloric acid, followed by drying. However, the extraction process used is determined by the chemical composition and characteristics of starch. Because of its low water content, extracting starch is much more challenging. Based on Mahajan and co-workers in 2021, starch extraction is broken down into three phases. The first one is fragmentation, then destruction of cells and collection, and finally purification. To provide improved extraction, the confined starch granules must be liberated from the protein complexes. Let us discuss biofilms from animals. The first one is collagen biofilm. Collagen is the principal fibrous protein from the skin, bones, ligaments, tendons, and cartilage of bovine animals and aquatic species such as fish, jellyfish, and sponge. These fibers provide the structural support to the body organs to ensure their strength and elasticity. This is necessary for effective locomotion, tissue regeneration, and repairing through the mechanical and chemical transduction processes. Collagen is generally colorless, opaque, and presents significant viscoelastic behavior by having a high tensile strength and low elasticity. Collagen molecules are made of 3 alpha chains associated in a triple helix matrix that are stabilized via intra and interchange hydrogen bonds. Polytrichoproteins are also considered as an interesting source for food processing collagen based high value products. Aquatic animals are the other main source of collagen and generally have different functionality than the terrestrial sources. Collagen can be isolated from animals via acid solubilization, pepsin solubilization, or supercritical fluid methods. Studies have shown that collagen can be used in biodegradable or edible films. Based on the fertilizing properties of hydrolyzed collagen, they suggested potential for use in agriculture applications. Scopel and co-workers in 2016 extracted collagen hydrolysate from chromed leather waste, which is hazardous waste, and used the collagen for preparing films. Dear listeners, let us discuss about gelatin films. Gelatin is a water-insoluble protein generated from moderate hydrolysis of collagen, either by chemical, enzymatic, or thermal treatments. The type of collagen source and the age of the animal are the key factors affecting the properties of gelatin. The amino acid composition of gelatin is generally glycine 27 to 30%, proline and hydroxyproline 20 to 24%, which closely resembles that of the parent collagen. However, conversion from collagen is accompanied by molecular composition changes in several amino acids. After the partial denaturation of collagen and during the cooling stage, the triple helix structure of gelatin that contains unstructured domains is formed. Gelatin is widely used as stabilizer, gelling agent, emulsification in food, pharmaceutical and cosmetic industries. Studies have shown that gelatin films have a high mechanical strength and barrier properties that make them suitable for food packaging application. However, the mechanical and barrier properties of gelatin films are highly dependent on the amino acid composition and the molecular weight of the polymer. The composition of amino acids, which differs between species and the processing conditions, affect the molecular weight and distribution of the gelatin. According to the United States Food and Drug Administration (FDA), FAO and WHO, gelatin is a safe additive for the application in the food industry and can be used widely in edible packaging. Now, let us learn about keratin films. Keratin is a general term for a variety of insoluble structural proteins which make up the main component in hairs, wool, horns and nails. Keratin proteins are rich in sulfur containing amino acids, giving them the ability to form disulfide bonds with other intra and intermolecular cysteine residues. The presence of disulfide and hydrogen cross-links in keratin proteins give them a crystalline structure, high resilience and rigidity. Each year, around 2.5 million tons of wool and 65 million tons of feathers are produced around the world. Wool is used in the textile industry and in small scale, low value applications, and the rest is disposed. The amount of cysteine from wool and feather sources are 11 to 17% and 7% respectively. Research has shown that keratin can be used for manufacturing of sponges, films, fibers, tissues and regenerative medicine. This is due to its biodegradable, biocompatible and biological properties. Different studies have shown that keratin films are commonly too weak and mixing glycerol in the keratin solution leads to better films with improved mechanical properties. Yin et al. 2013 reported that keratin films have a good mechanical properties and also pH responsive behavior, thus can be used as an excellent candidate for drug release applications in biomedical fields. Keratin films can be also used as a humidity sensor due to their low cost and rough surface morphology. The study of the effects of physical and chemical treatments on feather keratin films showed that the drying condition has the greatest influence on the properties of the film. Different chemical and physical treatments such as cross-linking by formaldehyde or soaking the films in isopropyl alcohol or weak acid improved the physical properties of keratin films. Keratin has also been used as biodegradable scaffolds, for example, in tissue engineering or developing 3D printing responsive materials. Dear students, let us discuss about biofibers. A biofiber is a fiber derived from biological origin, whether produced naturally or via regenerated process. Hemp is one of the oldest cultivated fiber plants and is currently finding renewed interest. Agro-based biofibers have the composition, properties and structure that make them suitable for uses such as textile, pulp and paper manufacture. In addition, biofibers can also be used to produce fuels, chemicals, enzymes and food. Byproducts produced from the cultivation of corn, wheat, rice, sorghum, barley, sugarcane, pineapple, banana and coconut are the major sources of agro-based biofibers. Using these crop residues for industrial applications could be an additional source of revenue for farmers without adversely affecting soil fertility. The next is fiber extraction. Natural cellulose fibers are extracted from lignocellulosic byproducts using bacteria, fungi or mechanical and chemical methods. Retting is the traditional process of extracting fibers which uses bacteria and fungi in the environment to remove lignin, pectin and other substances. Bacteria such as Bacillus are used in water retting, whereas fungi such as Rhizomucor and Fusarium species are used in dew retting. These species are found to be most effective in their ability to attack the non-cellulosic substances and separate the fibers from the core. Although atmospheric retting provides better quality fibers, but it requires relatively longer duration and it is difficult to control the fiber quality. The chemical retting methods use alkalis, mild acids and enzymes for fiber extraction. Sodium hydroxide is the most commonly used chemical for fiber extraction. Acids such as sulfuric acid and oxalic acid in combination with a detergent have also been used for fiber extraction. Chemical concentration, temperature and duration of treatment are the main factors determining the quality of chemically extracted fibers. For non-enzymatic fiber extraction, a combination of enzymes such as pectinase, hemicellulase and cellulase are generally used with a pre or post chemical treatment. Recently, enzyme complexes that can express 10 to 15 enzyme activities and provide better fiber quality have been developed. Mechanical separation of fibers is carried out using machines, steam explosion, Tilby process and similar methods. The Tilby cane separation process separates sugarcane into the outer skin, rind and sugar containing pith. The outer skin contains wax that can be extracted for use in pharmaceutical products. The separated rind can be used for the manufacture of paper, panels and boards. After extracting the sugar, the pith can be used for the manufacture of fiber, boards, absorbents and fillers. The Tilby process is stable for processing corn, sorghum and other lignocellulosic plant materials. This process offers the advantage of separating the plant components without disintegrating their components, which can be further treated to extract fiber and other byproducts. Cellulose fibers suitable for textile, paper and other applications have been extracted from wheat, straw and sugarcane by Tilby process and also by steam explosion. Rayon, the first regenerated fiber, became popular owing to the shortage of natural fibers, large availability of renewable forest resources and its relative low price. The most common rayon production uses sodium hydroxide and carbon disulfide to form a xanthine solution, which is ripened and extruded into a sulfuric acid coagulation bath to form regenerated cellulose. However, the development of cheaper synthetic fibers, environmental concerns and depleting forest resources led to the fall of rayon. In recent years, an environmentally friendly process based on the N-methyl morpholine N-oxide monohydrate NMMO solvent is being widely used for the production of regenerated cellulose. Now, let us discuss about fiber structure. The dimension and the arrangement of unit cell in a fiber determine the structure and also influence the properties of the fibers. The dimension of the individual cells in natural fibers are dependent on the species, maturity and the location of the fibers in the plant, and also on the fiber extraction conditions. Individual cells in pineapple and banana fibers are relatively longer, and therefore, these sources can produce longer fibers. The widths of individual cells in rice, wheat and corn husk are smaller and can therefore form relatively finer fibers. When used for applications such as textile and paper, the length to diameter ratio of individual cells in a fiber affects the flexibility and resistance. Wheat and rice straw have high length to diameter ratio and therefore more commonly used in the manufacturing of paper. Dear students, this is all for today. Hope you found the lecture interesting and informative. Thank you very much.